Okay. So if you don't have time, like if you're not quite finished with this, we'll have time during the break, or we'll probably have some time after I do all the things today. So what we're going to do today is wrap up this biotechnology chapter. Really, we're just going to focus in on that first, I think the first section in your textbook. So really, we're just going to, and then there's all these details. I'm not going to make you memorize all this complicated stuff that probably you're not ever going to need to use. So we'll just all hit the things that, like, all of these various techniques that are in common, and that's what we'll focus on. And then if you're interested in getting some of those details, and you have that available to go read about yourself. And then we're going to do the lab today. So I'll be, during the break, I'm going to go run and make copies for you all, but hopefully you guys read it over, so you just have at least an idea of what we're doing. It's pretty fun. We're going to be playing with micropipettes, which are so fun, but they're easily to break, so you need to listen and follow instructions. All right, so that's what we're going to do today. So let's do it. Okay, turn it on. Now push the button. Yeah. Okay, so we've talked a little bit already about Sanger sequencing, and we've talked about how I have used Sanger sequencing in my research over at NEU. And I just wanted to throw a couple more examples of how DNA analysis is used in the real world. So of course we use it for things like fraternity tests and stuff like that. But we also use it. So here's some, these are yellow shafted flickers. They're woodpeckers. And then we've got, and this is showing their distribution in Western United States and Canada and up into Alaska. And in the West, we have the red shafted flickers. And where they overlap, there's the orange shafted flickers. And so, like, are these 3 different species? Are they 2 species, with the orange shafted being the hybrids? Or are they all 1 species? We don't know. And so we can use DNA analysis to kind of sort out, like, okay, how close, we know what they look like and what their traits are, but like how close is their DNA? And then we can use this to figure that answer that kind of question. I've got this lovely little cladogram of all these cacti, so we can use this to look at how all these things are related to each other and understand the evolutionary relationships of these things. Here's a little bat with its, this poor little guy, he's got that, the white nose syndrome. This is an invasive fungus that kills bats. It's pretty sad. And it's spreading throughout the United States. It hasn't came to Arizona yet because it's too hot for that fungus, but we'll see if it adapts. But anyway, so we can use DNA analysis to track how this pathogen, this disease, this invasive species, is changing over time. And actually what we're seeing is that fungus is the same across all of its invasive range. So the Western United States, or the Eastern United States and South, and we can actually track how it's spreading, and we can see that, yeah, it's the same individual. So it's just the same clone, which is actually kind of good, because if we can figure out how to F with it, then we can eradicate it, hopefully. That's the hope. We also use it in forensics, as you guys probably know. We can find who the bad guy is based on, the saliva from the cigarette butt or whatever. Here's something that I didn't know that was happening. This is just a picture of a fur market, but a couple of years ago, I was taking a class where the students all had to select a paper, and one of the students selected a paper that was showing how we use DNA analysis to track the trade of endangered species. And so they can use either take samples of the meat or the fur or whatever the thing is that they're trading, to see if these are coming from places that were, or, you know, if they're poached or not. So that was pretty cool. Really interesting, like a law enforcement application that I was like, oh, that's clever. Of course, that makes total sense. All right, and then there's millions and millions and millions of more applications for DNA analysis. All right, so the basic things that we need to do in order to do any of these things is to be able to extract DNA or RNA, depending on what your question is. So I'm not going to go over that process because you guys have already done it, right? And so in your textbook, it tells you all that stuff. So if you're like not totally clear on it, you can look that up, or you can also look it up back on your lab sheets that you guys did. So once we get our DNA or RNA, we typically use this thing called polymerase chain reaction. And we just shorten it to PCR because that's easier to say. It was invented by this guy, Kary Mullis, who is so weird. Oh man. He got the Nobel Prize in chemistry in 1993. And then if you had a COVID test, you might have actually had your results come back using this PCR. All right, and then the next kind of the basic tools in the geneticist toolbox is gel electrophoresis, which is what we're going to do today, and then again, and then we'll finish it up on Tuesday. All right, so those are kind of the three basic steps. And you can do electrophoresis whether after you've done DNA extraction, before PCR, or after PCR. You can do electrophoresis whenever the hell you want. Whenever it makes sense for your thing. All right, so we're going to talk about PCR. Yes, I know. Okay, here's the memoir written by this Nobel Prize winner about this process that we have to like revolutionize the field of molecular genetics. And this is the cover. This is the picture that he chose for this thing. All right, that's a decision that you made, and the title is also excellent. So this guy, let me just read a couple passages from the first chapter. I was like, Jesus. All right. It gets better. All right. On the days when she still loved me, Jennifer, his girlfriend at the time, thought it might work. On increasingly numerous days when she hated me, my ideas and I together suffered her scorn. I got one more. So then Jennifer broke up with him because she's a crazy person. Yeah. There's a general place in your brain, I think, reserved for melancholy relationships of the past. It grows and prospers as life progresses, forcing you finally, against your better judgment, to listen to country music. Just, you know, really important stuff to put in your memoir about how you discovered this process. Love this guy. He's so weird. All right. And it's highly probable when he came up with this that there was some LSD involved. Yeah. But let me just also point out to you all the times that people take LSD and don't get Nobel Prizes is way more often. Just saying. All right. All right, so what do we use this for? We use it for everything. This is kind of like after you do your DNA extraction, this is the next step usually. So we can use it for figuring out what your genotype is. What is the sequence of your DNA? We can use it to make more copies of DNA that we want, like we talked about when last time when we were talking about the plasmid, and we're gonna put this is how we create and make copies of those plasmids. We can look for mutations. We can do sequencing. We can do all the things. All the things. Pick that back up. All right. Okay, so what the heck is PCR? Well, it allows us to make copies and copies and copies and copies. So many copies of a specific portion of DNA that always end up to be about the same size. And so this specific region of a DNA is important so that we can focus on a single gene. And when we do the electrophoresis thing, you'll see why it's important that they all end up the same size. So this is a chain reaction, and each time we run through one of these cycles, which will make more sense in just a second, we see this exponential growth of the number of copies. So we start out with one copy. Why are my hands shaking so bad? Start out with, I'm going to do the stick method. That's terrible. All right, we start out with one copy of our template DNA, right? And if you extract your DNA, you're going to get all the genes in that organism, right, from your saliva or whatever. All right, so in cycle 2, what it's going to do is pick that, focus on that single gene that we're going to tell it to look for. And now we got 2 copies, and then we got 4 copies, and then we get 8 copies, and then we get 16 copies, and blah, 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 blah. By the time we've done this 30 times, we have billions of copies of this one gene. So that's cool. All right. All right, so let's talk about the 3 steps that are involved in each one of these cycles. So the first thing that you have to do, so this is what Kerry Mullins figured out when he was on LSD, driving through the canyons of California in the 80s. And it was the 80s. LSD is what you did. It's for fun. All right. Step 1, the first thing that you got to do is you got to take your double helix and you got to melt it into 2 single strands. Okay, so we call this denaturing, which is slightly different than the denaturing of proteins, but yeah, right. All right, we're going to make 2 single strands. This usually happens close to the boiling point of water, so usually between 94, 97, 98 degrees. So not quite to the boiling temperature, but really close. And then the next thing that we've got to do now that we have our 2 separate strands is we got to take our primers and we got to anneal them or hook them onto our template DNA. So we've designed these little short primers that are correspond with this known DNA sequence. And we can design these primers. We can have a forward primer that's going from the 5' to the 3'. And then we can have a reverse primer that's going backwards, 3' to 5'. And then the annealing temperature that's required for this depends on the specific sequence of your primer. So if the regions that you're interested in, they have a lot of G's and C's, it's going to require a higher temperature just because you have to make more hydrogen bonds. And so this says 54, but really the range varies a lot, between 50 and all the way up to like 65 degrees Celsius. So pretty big range. And then if you're going to use both the forward and the reverse primer, you want both of them to have the same annealing temperature, or close to it, because that would be done with you. All right. So let's talk a little bit more about primers. So these are short pieces of DNA that are used to complement the template DNA. And so this is why it's nice to have that conserved region of the DNA, like we talked about when I was talking about how I look for fungi in plants. And we know that there's portions that are always the same across all of our different organisms. So we can say, okay, this is what our primer sequence is going to be, so that we can get this sequence in here in the middle that we don't know. Okay, so this is in preparation for where we're actually copying the DNA. And so we usually use forward and reverse primers, but not always. So you can if you want to just have the forward sequence or just the reverse sequence, and you can do that by just putting in the proper primer. Okay. And then the next thing that we got to do is the extension. And so this is where we're actually having the polymerase hook on to where we had our primer. So here's our primer sequence. The polymerase is like, yep, all right, here we go, smash. And now we're going to be incorporating new nucleotides into a new strand of DNA. Okay, using the same mechanisms in DNA replication, because that's exactly what we're doing. We're just doing it in a test tube instead of in our bodies. Okay, so you'll notice that we're going from like 94 down to 54, back up to about 72 degrees Celsius. So these are all happening at different temperatures because those temperatures are required for these particular reactions to occur, right? It's the ideal conditions for these chemical reactions. All right, so each of these, so one cycle has each of these 3 steps. So now we're ready to do cycle 2. And so our solution is about 72 degrees. What we got to do is pick it back up to 95-ish degrees in order to denature our double helixes we just need. Now, the first time that Carrie Mullis tried this, he just like stuck a bunch of stuff in the tubes and he like set it overnight and he was like, okay, bye. What's going to happen to our polymerase when we heat this thing back up to 94 degrees?
Thermophile, it wouldn't be fault, it just has to be a
Yeah, so this polymerase, and this is what Carrie Mullis was finding, is that those polymerases would denature. And so what he would have to do, and what they did for a long time, was that you would take your stuff and you would go, okay, so here it goes, put all the reactions in, all the things that I need to do to denature it. Okay, put it in this water bath, let it sit at 94 degrees for 2 minutes. Okay, ready, timer went off. Now we're going to put some more stuff in, put the primers in, put it in this water bath at 54 degrees. Okay, wait a minute or 2 minutes. Okay, timer goes off. Okay, now we're going to put in more stuff. Now we're going to put in our polymerase. Okay, now we're going to put it in this water bath and wait for 2 minutes. Okay, now let me go back here. Put it in the 94-degree water bath. Okay, now I'm going to put it in the 54-degree water bath. Okay, now I'm going to put it in. And so you can see that this would take a long time and a lot of effort and was really obnoxious. Because every cycle you would have to put in a new polymerase because it would get denatured at the next cycle. But then, there's these bacteria that live in the hot springs of Yellowstone, which is, hey, that's almost boiling temperature of water, just under. And these things live there. They ain't gotta have polymerase, right? For them to be able to reproduce and do the stuff. We can use this bacterium called Thermus aquaticus. We can yoink its polymerase out. We can throw it in, take its genes, throw it in E. coli, make it transform those E. coli to make this TAC protein, this TAC polymerase. And now we can do it for real cheap. And all we got to do is put all the crap in the tube one time. We'll put it in this machine called a thermocycler because it cycles through the temperatures. And then we just close it. We set enter the program. We close it up. Now we can go for lunch. So that's a lot easier than it used to be. And this is how I do it. I don't do the water baths. That sucks. All right. So this Thermus aquaticus really revolutionized how we do PCR. And then once we figured out how to transform the E. coli to make this polymerase, then it became hella cheap comparatively. It's still pretty expensive, but it's a lot cheaper than it used to be. Okay, questions about PCR? Okay. All right, so now that we have our PCR product, so we have copies and copies and copies and copies and copies and copies and copies of our gene of interest. Now we need, well, so first of all, what you do is you got to see if it worked, right? Because you put all the stuff in and you ran the thing, and then sometimes the universe is like, no. So we got to see if we have, how much PCR product do we have? Do we have enough stuff to do the next part? So how are we going to do that? Because so here's an example of what my DNA extraction looked like. It's supposed to be clear, but you know, there was all sorts of dirty shit in my samples. So how do I know? It just looks like a tiny tube of liquid. How do I know if there's DNA in there? Well, what I can do is make some fancy science jello. It's called agarose gel. And we're going to play with some of it later. And I can take a small bit of this and put it into a well that I made when I made the jello. And I can put it in this little rig thing that's full of buffer. And then my DNA sample is going to be attracted to the positive end of this electrode. And then I can like throw it under some shiny fancy lights and then it'll glow. So what it looks like, the gel photo when you're done with it, it looks kind of like this. So let's just remember that on our nucleic acids, that phosphate group, part of that phosphate sugar backbone that we know and love, it has a negative charge. And so that means it's going to want to go hang out with the positively charged end of our electrophoresis gel rig. Okay, so now I'm going to make you guys think for a minute. What are some of the factors that we talked about that affect the rate of diffusion? Temperature, size, viscosity of the fluid. Okay, so really what we're futzing with here is size. So the temperature of all of these things are going to, the rate of which our DNA is going to diffuse through our gel is all kind of the same. Charge matters, but they're all positively charged. One of the things that we didn't talk about is shape of the molecule is going to matter. So hopefully we have these nice little linear pieces of DNA after we have our PCR product. All right, so let's look at this thing, because what does that mean? Nobody knows. I know. All right, so here we have our wells. Whoop. All right, spit time. So these are the little wells that we made when we cast our gel, when we made our jello or science jello, with this little foam in there that left these little impressions. And we can drop our DNA samples into here. And then these ones are always reserved for a special thing called a DNA ladder. And a DNA ladder, oh, I think I have a slide of it. Yeah. Okay, so the ladders, you just buy them from Carolina or Fisher Science or whatever company. And they have in solution a bunch of pieces of DNA of known size. And so they're going to travel this. So help me remember, if you're a little molecule, are you going to go fast or slow? Fast, right? Yep. So, okay, if you're a little, you can slip through there easy. But if you're big, you're gonna be running into stuff. So it's gonna take you longer. All right. So this means, here's the starting line. What can we tell about this fragment of DNA? It's going to be little, right? Because it got farthest. And so then the ones up here, those are going to be the biggest pieces. And so when you get your ladder, they're like, okay, you run your gel under these conditions, and then it's going to look like this, and then you can kind of say, okay, so here's our actual samples. in these lanes. And so we can be like, oh, okay, so if this fragment of this DNA is probably about blah, blah, blah base pairs long, right? So then you can make sure that you got the right size in your PCR product that you wanted. The other information that's contained in this is how bright these bands are. So if they're really bright, then that means you have a lot of DNA. There's a lot of fluoresce in there. But if they're, so there's, this band has, there's a lot of whatever that size fragment is. But this one has very little. So now you can kind of get an idea. It's not a really like a way to quantify it, but it's definitely some qualitative way to say this one's more than that. Okay. Okay, so the other thing that I want to talk about before we start the lab is the use of model organisms in genetic research. So we hear a lot about scientists using mice to understand disease in humans or something, right? Why is it important to have these models? So I'll point out this one and then we can talk about that. So Saccharomyces, this is baker's yeast or brewer's yeast, it's the same thing. So it's just a single cell fungus. And we use it a lot in cancer research. What can a single cell yeast tell us about cancer in humans? It's so different. Yeah, so we can say, so is it ethical? So we want to know of what the trajectory of this particular kind of cancer in a human. Can I give you cancer?
No thanks.
I could, but it's not ethical, right?
Sign right here.
Yeah, it's fine. Just sign this paper, sign this paper, sign this paper, sign this paper. Thanks, bye. Right? So that's not ethical. But we can use this eukaryotic organism that has a lot of the same metabolic pathways as us. And nobody cares if we give those guys cancer because then we just throw them in the autoclave and we myrtleize all of them. That's fine. Right, but we can still understand what's happening inside our bodies because a lot of the same processes are happening in our bodies. We're both eukaryotic organisms. We use, so these are all organisms that have their entire genome sequenced. So the first thing that was sequenced was E. coli, right? That makes sense because it has a small genome. And we use a lot in models because we don't care about it. Rice is the cereal grain that feeds the most people on the face of the planet. And so we want to know about that. We want to know all its genes so that we can make more nutritious rice using fewer resources so that we can feed more people. This plant, anybody ever seen one of these before? No, you haven't seen that in nature because that doesn't exist in nature. This plant was created and bred specifically for genetic research. It has 4 chromosomes. We can mess with that really easily. And so a lot of what we know about how plants work is based off of this little thing. We can modify, we can find the gene that creates this particular plant hormone and we can turn it off. And then we can say, okay, well, what's going to happen to this plant? How does it function now that it doesn't have that gene turned on? And then that gives us an idea of how other plants use that same gene. And so black cottonwood was the first tree to have its genome sequenced. I just love cottonwood, that's why I put it on there. And then the other one that we use a lot is fruit flies. So there's a gene called the Hox gene that was discovered in fruit flies, which tells us why we think that humans that are more symmetrical are more attractive. We discovered that using fruit flies. And it's just like being symmetrical. And So the Hox gene is the thing that determines the symmetry, the bilateral symmetry in our bodies. So that's cool. All right. What else is next? Okay, I'm gonna stop there. We're gonna go to the lab. So take a little break if you need to. I'm gonna go make copies. So we'll meet back here in 10 minutes.